We are recording. Thank you, guys. Thank you to whoever reminded me. All right, so what is it?
A.
Oh, all of them are, yeah, all of them are monosaccharides except for which one?
A.
A. Yeah, glycogen. Glycogen is like I said, it's a complex carbohydrate made up of a whole bunch of glucoses. Remember, it's the energy storage molecule in animals, right? What's the energy storage molecule in plants? Starch. All right, good. Oh, did I not put the answer? I don't know, whatever. The cell uses a blank reactions to synthesize biomolecules. What's the name of the one that we use for synthesis?
Dehydration.
Dehydration, right? We're synthesizing, water's removed. Because think about it, when the two molecules join together, there's some atoms that are going to have to be removed to make room for that bond, right? When you're talking about hydrolysis and breaking them up, you got to add back some atoms to basically complete, you know, the valence shell of those molecules. I don't know if that just confused you, then don't worry about it. But if you kind of think about it that way, that might help. Okay. So bottom line is, when we talk about synthesis of biomolecules, the general reaction that occurs, right, is a dehydration reaction. Questions about that, guys? No, we're okay? Okay. All right, now which organelle does protein synthesis occur?
D.
Okay,
D.
this comes from chapter 4. All right, what about this one? So a protein that's going to be secreted out of the cell, right? It's going to be made on a ribosome that sits on the ER, okay? It goes into the ER, gets modified. Where does it go next?
C.
The Golgi,
D.
right? Remember we said the Golgi is like the sorting and packaging center of the cell, okay? All right, synthesis of lipids. What organelle do we have lipid synthesis? Smooth ER.
Smooth.
Smooth ER, letter C. Okay, where's chromatin found? The nucleus. What's chromatin? Yes, chromosomes are made up of chromatin, and chromosomes are kind of like a highly condensed version of chromatin. What makes up chromatin? You can just think of two things. Close, DNA, and then proteins. There's histones that wrap up, like the DNA wraps around, and that's the way it's packaged, right? If you kind of strung out DNA, it'd be super long and huge, right? In order to fit it into that tiny nucleus, it has to be packaged up, and we call that chromatin. So chromatin is your DNA, right? Your genes, and the proteins that are associated with it, okay? And when you think of a chromosome, it's a highly condensed sort of version of that, okay? Does that make sense to everyone? Any questions? No? Okay, so anyway, chromatin, right, DNA, is found in the nucleus, okay? Oh, Golgi apparatus is part of the endomembrane system. True or false, guys? True. So remember, all of the organelles that are part of the endomembrane system are made up of membranes, essentially. And they're all they're connected either via like vesicles or even directly connected, okay? All right, which part of a phospholipid is hydrophilic?
C.
D.
C, the polar head, right? So remember, we talk about a phospholipid, there's a phosphate group, this polar head, and two fatty acid tails. So what about those tails? Are those hydrophilic?
No.
Hydrophobic. Okay. So Vanessa asked kind of about this. So, you know, I just want you to understand that if something's hydrophilic, it means it likes water. It's going to readily sort of dissolve in water. Hydrophobic doesn't like water. So if you think about, you know, you kind of think about like an oil, right, in water, hydrophobic. I guess that's kind of a way to sort of conceptualize. When you talk about the phospholipids, right, the way they're going to organize themselves in the membrane has to do with this, right? So you have these two, so the polar head, right, which is hydrophilic, and then the two fatty acid tails, right? And when you, basically, if you're going to kind of draw, remember that I'm terrible at drawing, but you're going to draw a simple part, the membrane, right? You have a double layer. You guys agree? Okay? If you think about this being inside the cell, and this being outside the cell. Okay, so obviously it would sort of continue around, right? We can make a round cell or whatever, okay? Kind of going this way. What do you know about the, I guess, the amount of water, right, that's going to exist here inside the cell and also outside the cell? Do you think there's a lot of water there? There is, okay? So this is an aqueous environment, right? There's lots of water inside the cell and also outside the cell. So just based on the chemical properties of these phospholipids, that's why they arrange themselves in this bilayer, okay? This is the hydrophilic part that likes water, okay? Faces towards the outside. In this layer, that faces towards the inside because there's water here. These tails, which are hydrophobic and don't like water, face towards the inside of the membrane because there's, you know, kind of away from the water. So does that make sense? So the way, you know, a membrane, a plasma membrane of a cell sort of formed, kind of the basis of it, is really based on the properties, those hydrophilic and hydrophobic properties of those phospholipids that make it up. Does that help? Okay. Any other questions about that? Right. So I do expect you guys to know, you know, which part is hydrophilic, which part is hydrophobic. Okay. Yeah. All right. All right. Moving on. Okay. The nuclear envelope is composed of what? What'd you say?
C.
C.
Yeah, all of those
C.
other components. Yeah, right? All of those other components you guys say C here, right?
Yes, C.
The inner nuclear membrane, the nuclear pores, and the outer nuclear membrane. What are the nuclear pores? What are those? What do they do, I guess? Why are they there? Brad? Yes, and they let other things leave and enter the nucleus as well. Yeah, so they just basically are a conduit, right? Or provide an opening where things can get into and out of the nucleus. Brad mentions like the ribosomes, the subunits of the ribosomes, before they're sort of pre-assembly, they leave. There's also other proteins, nuclear proteins that need to get into and out of there, okay? So it's just you know, it's not just a hole, right? It's more elaborate than that, but it's just an opening to allow certain things to get through. All right. All right, I asked you this. So what's the answer to this question here?
Starch.
Starch. Thank you. Remember we said energy storage molecule, right? And I told you it's made up of glucose, so glucose storage, energy storage, okay? Same idea. Okay, all the following are biomolecules except for which one? Salts are not biomolecules. Okay, monomers of proteins. What are they? Amino acids, good. Okay, the monomers of proteins are amino acids, right? What are the monomers of nucleic acids? Nucleotides. Okay. Right, so remember again, that's all that same basic theme, the dehydration reaction, the hydrolysis reactions, monomers, right, polymers, all of those terms are general terms that you can apply to all those different biomolecules, okay? But then you gotta make sure you know the specifics as well. Okay. All right, prokaryotic cells do not have a membrane-bound nucleus. Is that true or false?
True.
True.
That is true. They don't have a membrane-bound nucleus. Do prokaryotic cells have ribosomes? They do. Well, a lot of single-celled, organisms are prokaryotes. Is that? I think that's what you're kind of trying to ask, right? Like bacteria. Yeah. There are single-celled eukaryotic organisms as well. Like yeast is a eukaryote, but it's single cell. Okay. But yes, they're single-celled. So bacteria are prokaryotes, right? They're smaller, they're much more simple. They do have ribosomes, though. Protein synthesis occurs, right? They lack other membrane-bound organelles as well, okay? All right. Oh, all right, so this kind of goes along with kind of an example I gave during the PowerPoint, during the recorded lecture. So it says red blood cells have a greater number of lysosomes than other cells in the body. Is that true or false?
False.
That's false. What was the example that I gave? White blood cells. Good. So why? Why does that make sense, I guess, if you think about the function of a lysosome and the function of a white blood cell? Brad. Yeah, we'll break it down. Say it again. What do you mean? So, well, correct. Red blood cells are small, and red blood cells are really only comprised of membrane, pretty much. There's not much else to them because the red blood cell's function is really to carry oxygen around, right? Hemoglobin is bound to it, and so they have a lot of surface area, and there's really not much else to them, okay? They're like cytoskeleton and membrane. White blood cells are larger. Specifically, the type of white blood cell we're talking about there it's called a macrophage. And what it does is it engulfs foreign material, right? So like bacteria, right? You get exposed to some sort of bacteria. These white blood cells can basically engulf that bacteria. And when they take it in, it then goes into a lysosome that has digestive enzymes to break it down, right? And destroy it. Does that make sense? Remember we kind of said the lysosome is the digestive system of the cell, okay? So think about a single cell. So in your body, you have white blood cells, part of your immune system, right? One particular type of them can do this. There are other types of white blood cells as well, okay? Questions about that, guys? You all right with that? All right. So the answer is false. It was, my example that I gave was white blood cells. Here we go, another cellulose question. Oh no, I didn't ask about cellulose, I asked about starch before. Anyway, what's cellulose? What type of biomolecule? Carbohydrate. And particularly, what type of carbohydrate? Moses, because you asked about this last time, I think. Remember you asked about structural, right, versus... Yeah, good. So cellulose is part of cell walls, right, in plants. Yeah, structural carbohydrate. But because we kind of divided those complex carbohydrates kind of up into sort of the energy kind of related carbohydrates, right, like energy storage, and then structural. All right. All right, saturated fats have double bonds. False. What type of fats have double bonds? Unsaturated, good. And what else? So what about saturated fats? Are saturated fats solid at room temperature or are they liquid at room temperature? Solid. So saturated fats are the ones that aren't so good for you, right? They're solid at room temperature, like butter, right? Lard. Unsaturated fats are things like plant oils, right? Like sunflower oil, olive oil. Those are liquid at room temperature, okay? And they have double bonds. If you think about the structure, right? I told you should be able to identify that structure from a picture, and really you're just looking for the presence of a double bond, right? But what does it do to those fatty acids, the double bonds? Yeah, it separates them, right? And that's why that molecule is liquid at room temperature, because there's more space in that molecule, more fluidity. Yeah, because of the double bond, okay? All right. Saturated fats have double bonds. False. Okay, which cells have a cell wall? Plant or animal?
Plant.
Plant. Do plant cells have a plasma membrane? They do. Still have a plasma membrane, but then a cell wall around it. All right. Okay, where does production of ATP? Mitochondria, right? We say the mitochondria are the powerhouses of the cell. That's where ATP will be produced. Right, so remember I told you guys you should just know basic function of these organelles. That's what I mean by it, okay? Okay, so what is this describing? What structure of protein? Primary, secondary, which one? Primary. So remember that the primary structure of a protein is just its amino acid sequence, okay? So why do we talk about proteins being different structures, or different levels of structure, I guess? Yeah, just think about it on a simple level, right? They need to be a proper shape in order to function, okay? So just having an amino acid sequence it's not functioning at all. It can't do anything. Has to fold properly, right? And then those other levels of structure describe those kind of different levels of folding, okay? All right. Okay, so we got them all right. All right, guys, so right now we're scheduled to have our exam on Tuesday, right? I don't know, how ready do we feel? Anyone feel ready, or do we feel like we need an extra day? I don't know, I mean, you know, we have to make sure we're kind of keeping up on, it is a lot of material, right? Those 4 chapters ends up being a lot. I think the first chapter is like, it's fine, but then once you kind of start getting into it, it is a lot of information. So, you know, I guess I'll think about it and I'll let you guys know, but be prepared. Your exam is next week. It's either going to be Tuesday or Thursday. Remember, it's delivered via Moodle, right? Nobody's going to be in the classroom. Everyone's just going to take it on their own. For the exam, there is no exception. Your camera has to be on. You have to be sitting at a computer somewhere, in a quiet place, and that's the end of it. I just gave an exam to 40 students. Not one person had an issue with being on their camera, right? Everyone took the exam like they were supposed to do, okay? So I will not make exceptions. For the quizzes, fine, whatever. But for the exam, no, okay? We're in college, guys, okay?
I have a question.
So just remember that, okay? I mean, I guess for those of you guys that are on campus, you could go to the computer lab or something like that, but I guess whatever you want to do. I mean, you could come in here and take it if you know it's going to be quiet. I don't care, all right? So just make sure you're somewhere quiet, okay?
Question.
So what was I going to say? Ooh, I forgot something. Oh, for the quiz, these 2 quizzes that we took, okay? And I'll send you guys an email about this. I will open them back up. I will give you guys all another attempt so that you can go in there and you can practice again and improve your grade, okay? You guys all hear that on Zoom?
Yes.
You said all the quizzes we took are the last 2. I'm sorry, I just came back in my room, so I didn't hear the last.
What'd you say?
All the quizzes we just took are our last 2. So
Well, there's only 2. The other one was a syllabus quiz. You did a chapter 1-2 quiz and a chapter 3-4 quiz.
Okay.
So those 2, I will reopen up, and I'll tell you guys, you know, you have between this time, you know, whatever, this day and this day to do it. You can go in, have a second attempt, and improve your grade and practice again, okay? And then you'll be able to view that attempt after that too, so that you can use it to study for the exam.
Is the exam going to be timed like the quizzes are? Because
Yep, for sure. The exam, you need to log on time for class, and it will be timed, okay? And it has to, and you know, I'm strict about the time because the idea is we can't spend, you know, hours and hours looking up answers. That's not the point. You'll have your notes there, just to kind of like jog your memory if you forget something. But you
our
have to
notes
be
don't
really
help
studying
because
for it,
we don't have
okay?
time to read the notes.
And this exam will also have essay questions.
How much time do we have for the exam?
You'll have an hour and 15 minutes, your class period time.
And how many questions?
It'll be about 40 multiple choice and like 3 or 4 essays, short essays.
And you said, just to reiterate, you said notes, we can use our notes, correct?
Yes, you can use your notes. Usually I'll give you guys, sometimes I'll give you guys, probably not. Anyway, it'll be about 3 to 4 essays. The essays will be more heavily weighted than the multiple choice, just so you know. So it may be smart to do the essays first, right? In case you're kind of running short on time, because they're worth more points, right guys? So you have to pay attention to the time. When you're in your quiz, right? You guys will see, I hope you guys are in your exam, it's the same setup as the quiz. You over here, where you have your navigation, you see your time there, okay? You have to pay attention to that. Any other questions about the setup for the exam?
Do you want the essays?
Say it again?
How long do you want the essays?
I can't hear you, sorry.
How long do you want the essays?
I'll tell you how long when I write the question in Moodle, but usually about 3 sentences or so. It's not like, they're not that long. But I'll say 3 to 4 sentences or 2 to 3 sentences or whatever in that question, okay? Other questions, guys? Okay. So for studying for the exam, obviously, you can listen to the recorded lectures again, or you just look at your notes. Go back to the quizzes, be familiar with those quiz questions. Remember, there are review packets that have even more multiple choice questions there. Go back to your homework questions. A lot of times there's, in the essays, there 1 or 2 of those homework questions shows up again, okay? Sometimes. So make sure you really review all of that stuff, okay, for the exam. All right, so let's pull up the chapter 4 lecture just to kind of hit on a couple of things from there. And then I'll let you know for sure whether the exam's Tuesday next week or Thursday next week, okay? If the exam ends up being on Thursday, then we won't have a live day, right? I'll let you know. Maybe I'll just switch the day, okay? I'll let you guys know about that. So I may just switch the day, depending on whatever day the exam isn't on, then we can be live that day, okay? For the review, essentially. All right, so let's pull up this chapter 4. I will tell you that, like, I don't really, I mean, I guess it could, there might be a question, multiple choice question or something like that, that might refer to this surface to volume ratio. But the idea is to understand, right? The whole point of this is like, why are cells so small, right? They're really small because if you talk about their surface area, right, versus their volume, the surface area is much greater, right? If you guys look, here, where it's showing you the surface area versus the total volume, right? So here, if you divide up this cube into 64 little cubes, right? You have much larger surface area, but it's actually the same total volume, okay? So that's the idea. The cell has to be able to sustain all the metabolic reactions that are occurring, right? And be able to get enough nutrients into the cell and then also be able to dispose of it. So it has to have a lot of surface area to be able to do that, okay? you know, I'm not gonna harp on this too much. That's why I wanted to pull that up, kind of like if you're, I don't know, confused about it or something like that. It's really the whole point of that, the take-home message, is to sort of understand why cells are small, okay? And the idea is that they have a greater surface area to volume ratio, okay? And that's what this is illustrating here. Actually, one of our labs down the line, the diffusion lab, does some more of this and kind of illustrates this a little bit more, okay? So it's not, again, for this exam, I don't even know if I ask you a question on it, right? Maybe one multiple choice question, but it's not something you need to study like a whole bunch, okay? Put it that way, all right? All right, if we move on here, I do expect you guys to, you know the difference between prokaryotic cells and know some characteristics of prokaryotic cells versus eukaryotic cells, right? I think we already kind of said this. So if I asked you to give me an example of a prokaryotic cell, what would be the first thing that came to mind probably? Bacteria? Yeah, right? Or archaea? Those are really the prokaryotic cells. And again, they're single-celled, like Vanessa had said before, okay? All right. So again, this is something like, I'm not on the exam gonna show you like a picture like this and ask you to identify what the shape of this prokaryotic cell is, okay? Sometimes we do this, well, we actually do this more second semester, but we'll do it on a lab practical, right? Because we'll look at slides and stuff like that. But for now, this is kind of just like a little extra sort of information in there. But these are, I guess, just be aware of these different shapes, but I wouldn't necessarily put a picture on it, you know, on the exam and say, identify this or anything like that, okay? So these are just different structures that you might see in prokaryotes, i.e. bacteria. You'll see bacteria are in these sort of different characteristic shapes. And that's one of the ways that we identify them, right? If you're trying to figure out what type of bacteria is infecting your patient, right? And you take a sample and you look at, say, a blood sample under a microscope, you can look at the shape of the bacteria, right? And there's other biochemical tests that you can do to determine what type of bacteria it is as well. And that's how we determine it, right? It's not like some magic. It's like you look at it under the microscope, right? You guys will do that in microbiology. So, we focus on eukaryotes. In microbiology, all you do is focus on bacteria and other prokaryotes, okay? And you do labs like what I just explained, right? Doing different tests and things like that to identify different types. All right. So, structure of the prokaryote plasma membrane, do you guys agree that it looks similar to eukaryotic membrane? Yeah? In what ways is it similar, I guess? So actually, I don't think there is, you mean cholesterol? I don't think there's cholesterol in the prokaryotic membrane. But what's there? What are those, what's that orange? Proteins. Right, so this is just showing you different protein molecules that are within that bilayer, which we see in eukaryotic membranes as well, right? So many phospholipids, just like we talked about. Okay, so very similar, right? Just more simple, okay? And same thing, I'm not gonna put up a picture of the prokaryote here and ask you to identify parts of it, okay? So if you're wondering, that's not part of the exam, all right? Okay, so same thing, I mean, again, just sort of we want to be aware of what prokaryotes have, right? They don't have a membrane-bound nucleus, but do they have DNA? They do, right? They have ribosomes, they have to be able to make proteins, okay? But their structure and the organelles that they contain are much more simple, okay? All right, so then most of the chapter is focused on eukaryotic cells, okay? So again, if you're kind of just looking at just basic differences, I thought I have a table. Yeah, right, so there's this table that kind of just puts them side by side, which might be helpful. All right? Right, so I do expect you guys to know the difference between eukaryotic cells and prokaryotic cells. All right. So, what about this? I think I kind of like mentioned this last class. What is this showing? So like, by the way, a lot of times on the essays, I might put a diagram or a figure that from your PowerPoint, and I think I told you guys this, and ask you questions about it, or ask you to explain what it's showing, okay? The essays usually aren't just about like, you know, list the 6 characteristics of life, especially if it's open notes. Those aren't going to be the kind of essays that I asked, okay? It's more of interpretation, understanding type questions, okay? So what is this, what's the whole point of this figure here? Again, you don't have to memorize the details, right? If I'm gonna kind of ask you about this, it's kind of big picture, I guess, your concept. What's the point of this? One of you guys on Zoom, what's the point of this thing? What do you think?
Showing the different phases of the organelles.
Say it louder.
The different phases of the organelles.
Okay. Go kind of a little bit further with it. I think you're kind of on the right track. You said the different sort of like phases of the organelles. Meaning if you think about like over time, right? Like through evolution kind of. Yeah. Wait, go ahead. What'd you say? Why do I not have volume here? One second, let me see if my volume's all the way up. Oh, it's not. Go ahead, somebody else say something? No? On Zoom?
Oh,
Yeah.
sorry, I was talking about.
Go ahead.
How it reproduces, I said, and like the different like paces.
Okay, so it's not really showing you reproduction, right? It's showing you over time, like evolutionary time, right? Like long periods of time, how eukaryotic cells essentially developed, right? Or emerged, or evolved. Does that make sense? So that's what this is showing you, that if you think about primitive Earth, right, and kind of origin of life, probably the first cells that came about were prokaryotic cells, right? They're more simple. Makes sense, okay? But then from those prokaryotic cells, you had eukaryotic cells evolve from them. So that's what this is showing you. It's meant to be a sort of theory to explain how eukaryotic cells evolved from prokaryotic cells, okay? Does that help? Does that kind of help make sense of this? So the two kind of organelles that it's focusing on are chloroplasts and mitochondria, right? So it's kind of showing you like how did they obtain their organelles? Because one of the main differences between prokaryotes and eukaryotes are these membrane-bound organelles, okay? So that's what that's showing you. Again, I'm pulling this up because I don't want anyone to memorize this. I mean, that's just pointless, right? It's just to understand that bigger concept, okay? I could ask you that in an essay question, right? Explain to me what this is showing, okay? And so if you look at number 3 there, where it says cell gains mitochondria, it's saying that this larger cell actually engulfed a small bacteria, an aerobic bacteria, which means they can basically do cellular respiration using oxygen, and then that little bacteria then became the mitochondria of that bigger cell. Okay, again, this is over time, right? It's not like some like instantaneous thing, it like ate a bacteria and then, right? But this is kind of over time, all right? I don't want to spend any more time on this, but I wanted to make sure I pointed that out, that it's not about memorizing what's going on here, just understanding the sort of bigger concept there, okay? Any other questions about that, guys? All right. Okay, so we already kind of talked about this, and on your quiz I asked you about this, this endomembrane system. So the way that the PowerPoint and the book is sort of organized is to divide these eukaryotic organelles up, right, by talking about the endomembrane system and then the energy-related organelles. Okay? So, I guess I already kind of talked about mitochondria, but remind me again, why do we consider mitochondria as an energy-related organelle? Correct. ATP is produced in the mitochondria, okay? That's where energy is derived from in the cell. We call it the powerhouse of the cell. Energy is produced there. What about chloroplasts? Why are chloroplasts, so obviously chloroplasts are only in plant cells, right? Why do we consider them energy-related organelles? What happens inside the chloroplast? Somebody on Zoom, what process happens in the chloroplast? What was this? Photosynthesis. And in photosynthesis, there's one phase where essentially the sunlight comes in, that energy, or the sunlight is basically converted into usable energy, right? So the plant generates energy for then another phase where it can actually make its own food, its own carbohydrate, okay? The other thing I want to point out is that plant cells also have mitochondria, right? They have mitochondria just like animal cells to produce energy for all the other things that are going on inside the cell, okay? For chloroplasts, the energy that's produced there is really directly for the production of carbohydrates, okay? For that process of photosynthesis. So it's not a, you know, one or the other, right? Plant cells have both chloroplasts and mitochondria. Animal cells only have mitochondria, okay? So just remember that. Plant cells have both, okay? Chloroplasts are there for the plant cell to be able to make its own food, right? The mitochondria is there to generate energy for all the other things that are happening in the cell. Does that make sense? It's kind of a nice way to think about it, I guess. All right. Okay, and so I did tell you guys that it's good to use these two figures to study from. I'm not going to go through them or anything right now because you can't see those words, but shows you at some point I will, on the lab exam, you will identify parts of a cell, right? Like I'll put diagrams up and say, what is this? For the lecture exam, I'm not going to use diagrams. I usually try to separate that up between lecture and lab. So it's just knowing the function of these organelles, okay? But I like these figures because it kind of has everything just all in one place, right? You just want to quickly know what the function of an organelle is, you know, study from this, okay? And then the same thing for the plant cell. Okay, so starts off talking about the nucleus, right? Remember we talked about the nuclear envelope, okay? What happens in the nucleus, I guess? Name some things, or, yeah, I don't know, name some things that happen in the nucleus. Not translation, but what does? And what else? What's another Asian word between before translation? Vanessa, you thinking of it? Okay. Transcription. Okay? So replication is making a copy of the DNA, right? Transcription is making RNA from DNA, okay? All those things happen in the nucleus. Why? The DNA doesn't leave the nucleus. The DNA is in the nucleus, okay? So there's processes that occur there, and then remember, things can get out via those pores, right? We'll talk about those processes later on. Don't worry about that right now, okay? Also mentions here the nucleolus, okay? So if you guys look at this picture here, so it's showing you here, it kind of looks like a smaller nucleus inside the nucleus. That's called the nucleolus. What happens in the nucleolus? That's where the ribosomal subunits are produced. So why does the cell need ribosomes, guys? What happens in the ribosome? Yeah, protein synthesis, right? But they're actually, those subunits are assembled inside the nucleus, particularly in this nucleolus, okay? So you should know the function of that, what that is. Again, it's just really kind of, for us now, know the organelle and just basic function, okay? You don't need to know all the details. Those come in later on, okay? For right now, we're kind of scratching the surface, right? Okay, so I think we already talked about this. Well, where are the where are ribosomes located? We got like 4 more minutes, but where are ribosomes located in the cell? Yeah, ER, remember we said rough ER is endoplasmic reticulum with ribosomes on it. They also can be free floating in the cytoplasm as well, okay? So you find them in both places. And so this is kind of what this is showing you. So by the way, so I feel like kind of, maybe this picture shouldn't kind of be here because there's more detail than we need to worry about right now. But basically it's kind of showing you a couple things that I just mentioned, right? It's showing you inside the nucleus there. There's the DNA, right? And then you see the purple mRNA being made. We said that was transcription, okay? And then that leaves the nucleus through a pore. And where does, and then what's happening here? What is this showing you? And then what's happening to the mRNA? Yeah, it's being translated into protein, right? We said ribosomes are the site of protein synthesis. That's what this is showing you, okay? But again, by the way, there's much more detail here than we even talked about, okay? So like, this isn't something that I expect you to know the details of either, okay? So, you know, but it's kind of nice to kind of see what's happening. But there's kind of all this extra stuff in here that I didn't really necessarily talk about much, okay? That's for probably like next year, okay? All right. Okay, then we talk about the endomembrane system. Okay, so remember it's the nuclear envelope, right? The membranes of the ER, the Golgi apparatus, and then vesicles. Okay, everything kind of is all made up of membrane. So again, this is showing you both the rough ER and the smooth ER there. Okay, so function, smooth ER. If I asked you basic function of smooth ER, what's one thing you would list? Synthesizes lipids. Okay, good. Rough ER, be careful because really ribosome is the site of protein synthesis, but the rough ER is where proteins are modified. Okay, let's go with that if you want to just pick one function, I guess. All right, then what happens in the Golgi? What did we say kind of as the sort of basic function of Golgi? Yeah, the sorting and sort of packaging, right? It's going to identify, and somebody I think said on Tuesday, like label, right, proteins, and then send them where they need to go in the cell. Okay, so this is kind of showing you. Right, so we should understand sort of what's happening there, what this is showing you, right? This is showing you the components of those endomembrane system, of the endomembrane system. It's showing you the nuclear envelope, the rough ER, the smooth ER kind of, but a little misleading, but anyway. And then it's showing you the Golgi, and then something being secreted. So it could be a lipid, right, that's synthesized in the smooth ER. It would then go to the Golgi and be sent where it needs to go. But you could also have a protein that was, say, synthesized on a ribosome here, right? And then that protein, you know, moved through to the Golgi, right? And then was secreted, okay? But that's the idea, something synthesized, right? It gets packaged up in a vesicle, and then it gets sent to where it needs to go, either released from the cell or somewhere else in the cell, all right? All right, so that's pretty much, we're out of time, right? Endomembrane, and then the next part of it, we talk about lysosomes and peroxisomes, and then the energy-related organelles, okay? So, guys, you know, we're gonna keep the exam for Tuesday, okay? So the exam is on Tuesday. Remember that Katie is available tonight, right, for group tutoring from 7:30 to 9:30. So check in with her if you feel like you need that. If you guys need more clarification on something, email me and let me know, right? We could always do a Zoom meeting on Monday if necessary or something, all right? I guess that's pretty much it, okay? So exam is on Tuesday. That means we'll be in person again on Thursday, okay? Same place, unless you hear different. I guess that's pretty much it. If anyone has questions, I'll stay on. If you're in lab, right, for the first session, 2:15 in Henderson Hall 113, okay, guys? Or via Zoom. All right, guys? So I'll send you an email when I'm gonna open up those quizzes too. I'll let you guys know, okay?
When did you say you were gonna open the quizzes?
I'll send you guys an email to let you know. I'll probably open it tomorrow, like till Sunday or something like that for you.
All right, thank you.
Yep. Yeah,
Professor,
yeah. What's up?
I have lab. I'm on my way, but it's cold. So can I wear my hoodie there or no?
Yeah,
All right.
Did you check my email? I won't be able to go to lab today, so I will be zooming in.
okay. Yep,
Right.
just log on to Zoom if you're not gonna be there in person.
But there's a sheet that we have to sign. I don't know. Do I have to like sign it in the app here or
You
or
have
whenever I
to,
go
yeah,
back
you're gonna have to submit it electronically.
today?
Yep.
How can I sign it? In which app?
I mean, you may have to print it out, sign it, and then take a picture of it or something like that.
All right, thank you.
Okay? Yeah.
Before lab, or can I just send it after lab before
Send it
12?
after that. That's fine.
All right, thank you.
Yep.
That's all?
Also, can you hear me? Also, if we go to lab, do we need our laptops?
Once I open it, I can see. Okay, all right. I'll let you know if there's an issue. Yeah. 1 second, guys, on Zoom. Go ahead. Oh, you know what? I'm gonna use the quiz for attendance. So you're good. So I'm gonna do this. Okay? Henderson Hall 113. It's on the first floor, not the basement floor. You know how you have to walk in the basement? You gotta go up one level. It's kind of like, where the hell are they? Oh, it's like, if you face Henderson, it's on like the left side of the building. So you walk up and then you're gonna walk across. Okay? Yeah, 113. Sorry. Who had a question over here on Zoom?
Me.
Go ahead.
I'm going to lab. Do I need to bring my laptop?
Not necessarily. No.
All right.
Professor, I have a question.
Sure.
For those of us who, like Moses, myself, and Alex, are we logging on to like the Zoom or like, in person, like lab at 5:30?
Yeah, during your time, right? That's what's on the schedule? Yeah.
Just wanted to make sure that's what.
Yeah, if you're out, I mean, what do we put? No, isn't it 5 o'clock or do we put 5:30? Regardless, whenever you're at a class, come over. You can start a little early if you want to. Okay?
Thank you.
Yep, you got it.
Wait, I'm confused. You said we have lab at 5.